Today we are going to start with the insulin, and we have to recall several things. Because here we have the possibility to see the receptor of insulin. Here we have the nucleus, DNA, the plasma membrane, and we count the insulin receptor. Here comes. Insulin receptor has a characteristic. Insulin receptor is different than the receptor of growth factors. Growth factor receptors are simple units that dimerize when they join the growth factor or the growth hormone. Remember, insulin receptor is completely different because insulin receptor has 4 proteins, 2 of them with an intramembrane domain and an extracellular domain that is the recognition site. And other 2 with an intermembrane domain and 2 intracellular domains that represent the effector site. Of course, we are seeing here the protein extended. The protein is not extended. The protein has a 3-dimensional structure. That means that they are used in that way. The 4 are not extended as we are seeing here, but they are used in this way. Well, then we will need the hormone. And the hormone is just insulin that is coming. When insulin is recognized by receptor, we produce the activation. Then it is a mechanism of the hormone enzyme receptor. It's a receptor enzyme mechanism. And it's a tyrosine kinase linkage. And this tyrosine receptor is at the same time a tyrosine kinase. That means it will catalyze phosphorylation of protein, including itself. Then through the action of this interaction, it will start to produce phosphorylation of the intracellular domain, and this multiple phosphorylation of the intracellular domain will produce a covalent modification that will allow it to recognize the IRS1 protein. Of course, here we are seeing coming the RAS protein that is just an internal membrane protein but not a transmembrane protein. And it is located on the inner surface of the membrane. Well, always GDP. Here we have another protein that we are going to see that we are going to be the activator. What are those proteins? And the SO57 and the growth receptor protein 1. Here comes insulin receptor substrate 1. And this insulin will phosphorylate it, multiple phosphorylation. And once it is phosphorylated, it leaves this active site, the IRS1 active, insulin receptor substrate 1 active, and we produce a trimeric structure with the IRS1, the GRB2, and the sum of 7 less. And that is the complex that will activate RAS protein. Then they will be displaced, will bind the RAS protein, and as in any G protein, the interaction of this complex with the RAS protein will produce the release of the GDP and to take GDP. The exchange of GDP by GDP. This exchange, this replacement of the GDP by GDP will activate RAS protein. And this RAS protein will produce the activation of the mitogen activated protein kinase cascade, coming or starting with RAS1. This complex, RAS1 active plus RAS plus GDP, will produce phosphorylation of MEK1, another of the MAP kinases. This MEK will produce the phosphorylation of more MAP kinases, and these MAP kinases will move translocated to nucleus, and in nucleus we phosphorylate different activators of the protein synthesis, activator of transcription. Remember that transcription requires to be activated in the eukaryotic cell. Then phosphorylation will produce the covalent modification of those transcription factors, activators, and when those activators interacting with enhancer region, we produce or we stimulate the messenger RNA synthesis. This messenger RNA will be translocated to cytosol, where the ribosomes will synthesize different proteins and cyclin-dependent protein kinase. And that will explain the action of insulin as a growth factor. But other are metabolic enzymes. And those metabolic enzymes will explain the long-lasting effect of insulin, but not the short-term effect. All those proteins that we are seeing here, of course,